Good afternoon, one and all. Let us welcome Srimati Papia Bhadra, serving as CGT Bio in Atomic Energy Junior College, Mumbai. Madam has completed MSc degree in botany with specialization in microbiology. Madam qualified next CSIR for lectureship December 2000. Diploma in software technology. Madam had been working as PGT biology in Army Public School, West Bengal from 2002 to 2006. After that, Madam joined Indus Universal School, Yapral, Hyderabad from 2014 to 2016. Madam joined Atomic Energy Education Society in 2016 as PGT Biology, and since 2018, Madam is working as PGT Bio in Atomic Energy Junior College, Mumbai. So let us welcome Madam once again, and I hope you will enjoy her session and learn a lot. Thank you.
Thank you so much, sir. Good afternoon, students. Nice to see a bunch of students who are eager to learn things beyond their syllabus. Uh, I guess most of you are from class 9th and 10th level, right? Yes. Okay, so you have a basic knowledge of blood, right? Am I right? Yes. Okay, basic knowledge of blood in the sense you may have studied about blood as a tissue in class 9, where you may have learned about what is what is its composition. Blood as a tissue, what does it contain? Like RBC, WBC, platelets, this much, right? Plasma, of course. And anything else? So we, platelets, right? So we will go a little bit beyond that level. Okay. Have you ever wondered why some people will always say we are in requirement of so and so blood. Can you please provide? We want only B positive or B or A positive or O negative or what, sir? Yes. So now comes the point you are saying that is related to antigen and all. So we are slowly getting into our topic. Our topic is related to blood transfusion, blood as cell, as tissue. These things are known to you, and rest of the whatever things which you need to know, you will be studying it in your syllabus. But we will go beyond the syllabus level, right? So we will be basically dealing with today three things together. First, what is called as ABO blood grouping system. Then we will consider what is another level of blood grouping, which is considered as RHD system. Okay. And the third thing we will discuss is how genetics is involved in this entire topic. Okay. Where is blood and what is genetics? Genetics is not at all something which is you don't have basics of genetics so far. I know. But I will teach you what are genes and how genes are associated with this blood grouping. Right? So let us begin. First and foremost, the word transfusion. Transfusion means how the blood can be given to another individual. One individual can give or take blood and survive in dire situations. In situations where there is a person is dying. Say for example, there is a war-like situation or an accident, a train accident. There are many people who require blood. And sometimes in certain surgeries and all, you may require one patient may require blood. So at that time, that patient is given the blood from another individual. But there are many conditions related to that. Okay, anybody and everybody cannot give blood to a person. Only perfect match has to be there. That is what we are seeing here is what is the perfect match that will be. When we learn about that, then comes the word transfusion. When we are doing the blood transfusion, we need to see the perfect blood group match. So for that, before 1901, this was not known to people. Okay, at around early 1900s, this thing was coming to a picture, clear picture that blood has to be perfectly matched before it is given to someone or donated to someone. Okay. So in 1901, who was the person who brought this into light? Yes, he was from Austria, and next time. This is the person you can see, Karl Landsteiner. He was awarded Nobel Prize also, as you may have seen in the previous slide. He was awarded Nobel Prize because of his outstanding, discovery of how blood is transfused based on blood match, perfect match. So for that, we will now get into the details. Next slide, come. So this is what is the basics regarding blood. As such, any adult human being should contain around average 5 liters of blood. 5 liters? Any idea what should be the 5 liter? You may have seen 5-liter bottles, cans, water bottles. Yes or no? Or you can see simply, the same. Show me that bottle. This is roughly going to be. This is roughly going to be having 1 liter water, right? So this kind of 5 bottles will be 5 liter, and such 5 liter of blood is there in every single human being at a given particular time in an adult human being. So this 5 liter of blood is too much, yes or no? This 5 liter of blood is also sometimes not enough to support a life when, say for example, the person passes a lot of blood out of his body, for example, in case of accident or a surgery. So in that case, what happens? We need to provide the blood. Why? Because this 5 liter of blood is at a particular given time highly required to make that person survive. Otherwise, the heart will not work. 5 liter of blood should pass through the heart. Okay. So this 5 liter of blood is not available. Suppose the volume is little less, the heart will collapse. So at a given time, a person cannot survive with less amount of blood. Got me? So for that, what happens? We need to know. This is already known to you. Red blood cell, white blood cell, platelets are the basic things, and their function is also written in a very, because this is not a major topic right today. What is the function of the various cells of the blood is not our scope of study right. So we want to just show you this just to make you understand if somebody suppose cannot recollect for that check. RBCs are basically required for transporting oxygen and CO2. WBC is basically required for infection fighting infections. They are the soldiers of our body, as you all know. And the platelets are basically for clotting the blood. Suppose if the blood is coming out of your body, then after a while it will clot, it will club in one place and it will no more come out. So that is called as blood clotting. For that, platelets are required. So these are the 3 major things present in the blood. Apart from that, the blood contains a lot of proteins and many other molecules. That is present in the matrix called as plasma. The watery matrix is plasma. So this is all about the composition of the blood. So here, when we consider what is blood and what is the type of a blood, suppose if a person is blood type A, B, AB or O. These are the 4 basic types available, right? What are they? I repeat, A, B, AB and O. These are the first 4 basic types, main types. I am not saying that ABO blood grouping is the only way of grouping blood. There are many other ways also. Bombay blood grouping, MN blood grouping, many more are there. But that is not our topic today. So our topic is ABO blood grouping. In that ABO blood grouping system, you will find when we are telling a particular type of blood is A, on what basis we are saying it is A. There should be some reason, scientific reason. So before coming to this particular slide, I will come back to this slide later. And next one. Can you see now? This is the type A. As you can see, type A. Suppose RBC, red blood cell. All these types are based on RBC's structural feature, not WBC, not platelets. WBC and platelets are not involved in this matter at all. All the time when we are saying A, B, O, we are only thinking about erythrocytes, that is RBC. So suppose this is an RBC. RBC is plasma membrane is this. Okay. In the plasma membrane, on the plasma membrane, there will be a particular type of structure. Say for example, the structure looks like this. Okay, so what is this? Antigen. Why antigen? Because antigen as such should be considered or defined as something which is foreign in origin. Antigen's real definition is something which is foreign in origin. It can be a virus, it can be a bacteria, it can be any toxic substance, it can be even a pollen grain also, it can be a dust particle, it can be mite, it can be anything which is not the individual's own cell. Anything which is foreign in origin is considered as antigen. Did I make myself clear? If you have any query, let me know. You stop me. You can stop me. Just raise your hand. I will stop. Okay. Without making you really understand the matter, I should not go ahead. So if anybody doesn't understand in a particular point, please let me know. So antigen is something which is foreign in origin. So, but when I'm saying that RBC is containing this particular type of antigen, what do I mean? RBC is my RBC is somebody, some human, any particular human being's own material, and on that cell membrane we are having antigen. We are saying again in the second word I am saying sentence I am saying that antigen is foreign in origin. So am I contradicting myself? If I am, why am I? So the thing is, these antigens also are called as antigens. Maybe if you go millions of years back, you may see that prehistoric human beings may have taken it, or maybe if you see the evolution of human beings, you will find that these antigens were not present in the blood first. Later, it may have come through some other organism or through some other animal. It may have come long back. So actually it was foreign in origin. But later when it came to our system, we accepted it as okay, fine, let it be here. We are not having any harm with that. We don't have any problem with it. So such antigens are therefore still named as antigen, but which are not harmful. Okay? They are there, just simply there to give some identity to your special type of RBC. Means you are having an RBC with a special type of antigen in it that makes kind of, like you are you, he is he. How do we understand? Because you look different, right? Am I right or not? So this gives a separate look to the RBC. Got me? So for example, say for example, this kind of antigen, let us call it A antigen. Okay, let us call this A antigen. Then this kind of RBC, we will call this person as having A type blood. Okay, this RBC which has got A antigen on the plasma membrane, we will call it as A type blood. Similarly, B type blood, B type of blood will have. Let us consider this another kind of an antigen, which is B antigen. This person is containing all the RBC of his body having B antigen. Correct? Yes or no?
Yes.
Fine. What about AB? What do you think? What should it be?
Ma'am, both A and B.
Yes. So he should be containing both this and this, right? A and B antigen both will be there in almost equal amount on the surface of the RBC. Right or not? Therefore, we are calling it AB. What about O blood group?
No antigens.
Will have no antigen at all. So it will be devoid of any kind of antigen, whether it is A or B or anything, or AB. Okay? I can see some students are smiling. May I know the reason, please? You might be thinking something. Let me know. Anything funny? Up so far? Okay. So A. Yes. We will come to that. Just have some patience. Okay, A, B, AB, and O. Why we are calling such people like C? Here the slide you can see. A type of blood is having rounded antigen, right? B type of person is having a conical type of an antigen, and AB will be having both, and O type of person will be having no antigen at all. Got it? Yeah.
Antigen A actually round, or is it just?
No, it is for your understanding purpose, we have shown it in a particular structure. Huh? Shape is there. Of course, shape is there. That shape is decided by a particular protein called as agglutinogen. Okay, that protein shape will decide the shape of the antigen. That was a nice question. Anything else? Agglutinogen. Okay, that will come in a slide. I will write it for your understanding. Okay. The spelling is a little difficult, so. This is the type of protein which remains present on the surface, and depending upon the structure of that protein, the A and the B antigen will be separate, right? Now, this A, B, and O, okay, fine, it is there. But what else is also there? Along with this antigen in the plasma, blood plasma. Plasma, you remember we talked about plasma just a few minutes back. What is plasma? Plasma is the watery part of the blood, right? Not the cell. Fluid part of the blood on which the cells are present, right? On which the RBC, WBC, and platelets are present. In the plasma, you see a protein. As I said before, also plasma is loaded with many proteins. One of the protein is called as antibody. So, what is the location of antigen? I repeat, antigen is located on the surface of the RBC. And what is the location of antibody? Antibody is located in the plasma. Right? These antibodies are also very, funny. They are present in a separate way. Now, for the person who is having A antigen, what kind of antibody you are going to expect in the plasma of that person? B. But then when you are saying, since it is written, you might be saying there, but we need to have a logic behind it.
The antibody kills or
yeah, almost to the point. So let us understand it again, okay? Suppose this person, we are talking about a person who is having A antigen, and hence is considered as A blood group for a man. Okay, a person with A blood group. Why A blood group? Because A antigen. But what happens in this plasma? There are antibodies. Antibodies are particularly Y-shaped. These are proteins which are Y-shaped. And with Y-shaped protein, they attach to the particular antigen and they make antigen-antibody reactions. But in this person, if there is an antibody, say for example an antibody which is if consider such a person with antigen A, antibody is also say for example A, then what will happen? A antibody will combine with A antigen and the RBC will be destroyed. So such a person cannot have A antibody. A antigen containing person will not have A antibody, rather because A antibody रहेगा तो A antigen के साथ react करेगा. So as a result, he will contain B antibody. B antibody will not be able to detect A antigen and there will be no reaction. As a result, such a person is supposed to contain B antibody and not A antibody. Could I make this clear? So a person with A antigen will contain B antibodies. So we will see that on the slide, anti-B antibodies are present on the plasma of a person containing A blood group. Done? Is there anybody who did not understand? Similarly, if you have understood this, now it becomes very easy to explain about B. What about B? What will be the antibody type in B blood group person? Obviously, this antibody will be A antibody, right? Because if had it been B antibody, then B antibody would have reacted with B antigen and the RBC would have been destroyed. So ऐसा तो होता नहीं है. So as a result, the person will be having in B person A antibody will be there. Similarly, going ahead, what do you expect in AB blood group person? No antibody, because have no antibody. Antibody, let me write it as AB. So if a person is having AB blood group, if he would have got A antibody or B antibody, then it would have reacted with these antigen, then his blood would have destroyed. Would have been destroyed, RBC would have been destroyed. So that is not the case, so he will be having no antibody at all. But in case of O blood group person, yes, both the antibodies will be present in such people. Means both A antibody and B antibody will be found in person who is having O blood group. Is there anything which is not understandable in this? This was easy, pretty easy. I guess everybody has followed, but still, if anybody has not, let me know. I will pause for a while. Yeah.
But it's visible to disappear.
Let me. We will come to that also. See what happens. Since he has asked and it is written on the board also, on the PPT also. Let me explain. Suppose now, based on this understanding, if I say a person with A blood group is giving blood to a person with B blood group, what would be the repercussion? What would be the result?
The person will die very fast.
One by one. Can I give them? Yeah, please.
Antigen E would react antibody
Antigen B would react with
A.
antibody B because suppose A is giving blood to B, A is also giving B antibody. And B antibody will react with B antigen. And there will be a reaction. So what happens is, if this is the case, if for example this is A antigen and this is a specific A antibody, antigen will recognize each other and will combine with each other, they will bond with each other. As a result, these antibodies will attack on the cell membrane of the RBC. As a result, there is a reaction called as agglutination. So, biology is all about learning certain terms also, which is unavoidable. We cannot do anything about it. You have to learn, and once you start learning these, understanding these words and their meaning beneath, you will start loving them. It's not that such words are going to, make you, go away from the subject. So, agglutination is a type of reaction which happens when a specific antigen matches with their counterpart antibody. Say, for example, A antigen matching with B antibody and reacting with each other, they will make agglutination reaction. As a result, if there is such kind of agglutination reaction, what will happen, the blood will become curdled. How do you see it? If suppose if this happens in the blood, what do you see in the blood? How do you understand that the blood is having agglutination reaction going on? You will see the blood has become curdled. Now, what is the meaning of the word curdled? This is an English word. This is not biology word. English. Curdled. Clubbing, curdling. I'll give you a best example is when you see milk has sometimes you see that the milk is no more good at home and it just curdles. Or if you want to make paneer out of milk or cottage cheese out of milk, so you will put some acid into it, say some citric acid or something, and it will curdle, right? Same thing will happen to your blood. Blood which is having its own consistency will no more be of that consistency and it will become simply curdled. So that is called as agglutination reaction. So such blood is of no use, that will not come through your heart and your heart will not pump the blood to various parts of the body and as a result the person will die. This is why it is important to know the blood biochemistry, blood genetics is going to follow. So, have you understood? Now, the question was initially, why it is called universal recipient and universal donor and all that. Let us come to the next slide now. Now, we have discussed already the blood type A, B, AB and O. Genetics, we will come later. First of all, fourth type of blood that we have understood. Next slide. We will come to that later. Now, regarding universal donor and recipient, let me tell you once and then I will get to this. Say for example, A blood group person we have understood this, that A blood group person can take blood from A only, right? Similarly, B can take from B only or give to B only. Similarly, A can also give to A only. Giving and taking can happen within the same group, right? Understood. Second thing is, similarly, AB can give to AB or take from AB, O can take from O or give to O. But there is one thing more. Don't you think this AB can take from A also? Yes or no?
Yes.
AB can take from B also.
Yes.
Suppose in situation where AB matching blood group is not available and the person requires the blood immediately. Okay, what will happen? He will not, we don't have time, we don't have any option left. Say for example, we have only a person who is a person who is only having B group, or a person having O blood group. Can we take from him? AB is not available, suppose.
Yes.
AB donor is not available. So what will happen? We can take from A for a while. With some risk, I understand, because A means you are also inviting B antibody. And this person is also having B antigen, so those B antibodies will react. But still, half of his RBC's cell membrane will still be protected. Some B will work for agglutination. B antigen antibody reaction will happen definitely, you cannot avoid, but mostly it will be still okay, better than time. Right? Similarly, if our O group is O person is there, okay, can we take blood from A, B, or B? O blood group person, think. O blood group person is there. Can we take from A, B, AB? Yes. What will happen? Suppose he is having no antigen. He is containing all the antibodies. Even if he takes from A, what will happen? A antigen will be immediately attacked by A antibodies. Right? If he takes blood from B, his B antigen will be immediately attacked by the person's own antibodies. Am I right or not? So there will be all whatever blood we take, and if he takes from AB, consider the case. If he takes blood from AB, both A and B will be attacked by A and B antibodies because he is containing both the types of antibodies. As a result, this kind of a person cannot take from anyone. He can only give. So he is universal donor. He is only able to give blood to anyone. He can give blood to A also, B also, AB also. No problem. Because he does not contain any antigen. Okay. So for a while we can, best match is always same to same. O to O, B to B, A to A, AB to AB. That is the best possible concept. Best possible match. But if that is not possible, we are going for the second option. Then in that case, O is considered as universal donor. You can just come chill out for a while. You can take O blood group and can start your job. But and similarly, AB can be considered as universal acceptor or recipient. He can only take. He cannot give blood to anyone, owing to the fact because he is containing both the antigens and no antibodies. If he is giving to anyone, there will be antibody attacking on him, on his blood. So the thing is, still a question can come from you, saying कि अगर थोड़ा सा अटैक हो रहा है तो हम लोग फिर भी क्यों दे रहे हैं? हम लोग उसकी जान को उस पर्टिकुलर समय में बचाने के लिए दे रहे हैं. थोड़ी देर के लिए वो सर्वाइव कर जाएगा और हम लोग we can buy some time with that. At least we can make the process survive for a while. Anyhow, let me tell you, whether you are giving to the matching person also, say O is giving to O, A is giving to A, B is giving to B, then also what is going to happen after a while, do you know? The donated blood, yes, you want to say?
Since you said that like AB antigen person can take blood from both, won't the AB antibody attack the AB antigen?
Come again, AB?
You said that the AB antigen person can take blood from O.
Yes.
The O person has AB antibody.
This is a correct thing. As I said, his own blood will be, you are taking less amount of blood, right? His whatever majority amount of blood is still there to support him. But right now you want the RBC to do the main job, that is oxygen and carbon dioxide transporting. That has to be carried out. So for that particular purpose, you are taking against your wish. You are taking. Okay. Some amount of blood will be destroyed definitely, but to some extent, some amount of blood will still do the job. For the time being, you can buy some time and you can make the person survive. If there will be some attacks, that is unavoidable. As I said, unless it is completely matching, it is not advisable to take from these people. Except in war-like situation, accidents, we go for other groups. Otherwise, no. Okay. And as I was telling you, even if it is same blood group, then also there is a situation after a while the blood will be destroyed. The one who is donating the blood, his blood will be destroyed. His all RTC, WBC, everything will be destroyed in the recipient's body. Why you know? There is an immune system in our body which will recognize after a while that this is foreign in origin and it will be definitely destroyed. Even if it is of the same group. But then still we give 5 so that for that particular time being the person can survive. Am I clear? Finally it is also going to get destroyed. Those blood cells which are getting donated will be destroyed. Anything foreign in origin will be identified and destroyed.
Where do positive and negative facts fit into this entire?
You have to have some patience for that. We have not come to that topic yet. I am still on the ABO grouping only. We will have to wait for a while.
Ma'am, I have a doubt that if an O positive person, O type person is needs blood, but there is no O type person, so what will it do?
Since he is universal donor, so he has a little problem. He cannot receive blood from any other type other than his own type. And that is why good thing that nature has a balance, you know. Nature has made O people more in this world. So that is also a good thing, good news for these people. That many people are O. Okay, most and it also again depends upon race to race. Okay, many race in the sense, when I say race, you know, it is very risky because nowadays if you say race to race and this race has this race, that race has that, then people try to judge you. They think that okay, you might be racist, you know. So it is very risky to say this, but still it is there, which is true, you know, for this reason only. You know about Watson and Crick? Have you heard of their name? J.B. Watson and Crick, FNC Crick. Have you ever heard? In what context? Watson and Crick are the, you know, any person who is studying biology should know their name. They are the, you know, because these are the two people, those who gave the world the best gift ever possible. That is, they unfolded the structure of DNA for the first time in the world. People came to know about how the DNA looks, what is the structural detail of DNA in 1953 because of these two people. Okay, and out of these two people, J.B. Watson is still alive, and he, being so, you know, having so much of wisdom, he still had to face the problem of, you know, this judgment of, you know, as I said right now, because he said, because he is into DNA biology, so he said, so and so DNA will contain more, you know, this kind of DNA will have more intelligence, you know, these kind of genes are there in the races where they are having, you know, these kind of people, those who belong to this so and so race will be having so and so genes, and so genes means more intelligent. So such people are more intelligent if they are belonging to this particular race. And immediately he was attacked, saying, okay, so you want to say that we people, those who are not of this race, are dumb. So that's the case. So it's very difficult to say that way, but anyhow, this DNA has got something to do with race. So we are right now telling that Indian people are all mixture. As you know, we had survived so many invasions in history, we know that. So we have mixed blood, all the wait, let me complete this. So what happens, we will have mostly, many people will be having O blood group. As such, we don't have to worry about that. Now you can ask me. I will definitely give you a chance. Maybe.
What's golden blood group? Golden blood group.
There are many such. Golden, Bombay, we are not going to that because I know a lot of things. We will discuss it. I personally tell you.
Because my question was that if an O-type person needs blood immediately, but there is no O-type person to give him blood.
That is a trick. We cannot do anything about that. We, O type of person cannot take from anybody else.
And he wants to be right, he will die.
He cannot take. If he takes, he will be having blood mismatch because of agglutination reaction and he will not survive. That is for sure. But I have told, the O blood people are moving.
So then he will ultimately die.
So can we move ahead? So now we will discuss this now. We are now into the genetics part. We have understood the basics of ABO blood grouping. Have we? Yes or no? Now we will be understanding the genetics behind it. Okay? How this blood group is connected with genes. As such, we all know, I know that you people have not been taught about DNA, chromosomes and genes much. Just to give you the basics right now. See, we all human beings, can I have your attention please? So we all human beings are made of crores of cells inside our body. Yes or no?
Yes,
Thousands and crores of cells we have in our body, right or not? But do you know that all these cells have come from one single master copy? Like we do Xeroxing, right? Nowadays, we take one paper and we make hundreds of question paper out of it. Single question paper से 10, 50, 100 question paper बनाते हैं ना? क्या करते हैं? Xeroxing करते हैं ना? We do photocopying, right or not? Yes or no?
Yes.
Similarly, we in our body also have a very nice photocopying machine. How? We have first one single cell called zygote. Okay? One zygote is first made, which is diploid. Now, what is the meaning of the word diploid? Diploid means 23 pairs of chromosomes. 1, 2, 3, 4, 5, 6, 7, 8. Okay, 23 pairs of chromosomes are there. So if it is in paired condition, we call it as diploid. Understood? Yes or no?
Yes.
And if it is only 23 chromosome means sperm and egg. Only in our body we have one. If ladies will have one egg cell which is haploid, and men will have a sperm which will be haploid. When they fuse together, we get a zygote. This zygote is our master copy. And from this zygote, master copy, we make thousands and thousands of cells, which are all same. That means, if zygote has got this 23 pairs of chromosomes, we are all the time making 23 pairs of chromosomes as many times as we want. Right? Yes?
Yes, ma'am.
Whatever is there in these chromosomes in the zygote, we will have the same copies in crores of cells in our body. If any defect, all the cells will be defective. If any good thing is there in the zygote, all the cells will be having that good thing. Right? Now, what do I mean by defect? Would say could not get you. Mutation. Yeah, mutation, but that is not the thing which I want to know now. In chromosome number 9, say for example, we have chromosome number, say for example, this is chromosome number 9. Okay? In these chromosomes, we have genes. Okay? In one particular place, if there is a gene, the same gene will be repeated in this chromosome also. Right? So every gene has 2 copies. Say for example, now what do we mean by genes? In simple words, if I put, genes are going to express into proteins in your body. Whatever proteins you have, they are all coming from your genes, and genes are present in the chromosomes. And that too in pairs, because one is coming from mother and the other is coming from father. Right? So you are getting every gene in copy in a pair. Now what is that? Let us understand with example. What do I mean by genes and how do I really see it in our daily life? Say for example, I'll give you one small little task. Do this. All of you, leave your pen, put your pen aside, put your pen aside. Just simply, randomly lock your hands. Randomly, without thinking, lock your hands. Lock, keep it locked. Now you see, which is coming first, right hand thumb or left hand thumb? Now I want you to raise your hands, those who have right thumb, like mine, right thumb is coming first. Raise your hands, those who have right thumb coming first, see? Only few. And keep your hands down, left thumb first. See? Reason? Never thought that. Okay.
Because we did it without requesting the hand.
Yeah, I'll come to that point. You cannot do otherwise. See, even if you want, you cannot do because it is genetically governed. Many characters in your body is genetically governed. Hey, please be calm, quiet. Back. One, two more examples. Try to roll your tongue laterally, sideways, like this. Try. Many of you will not. How many of you could not? Raise your hands. Many of you could not. See? But many of you could. Right? See, you might be thinking, इतना simple चीज क्यों नहीं कर पाया? You ask them 100 times, they cannot. Those who cannot, cannot. Because these are genetically governed. You cannot do anything about it. Your thousands of cells are having that genetic code in them to determine whether you can do or whether you cannot do. Right? Some more examples do you want?
Yes, sir.
Oh, this is nice, right? We are drifting away from our main topic, by the way. Anyhow, since you like, I will be going along with you. So see, touch your ear lobe. Can you see the ear lobe? You will find some people are having ear. Suppose this is your chin. Some people will have ear lobe like this, and some people will have ear lobe just like this. Some will have lobe, some will have attached. Some will have lobe like this. There will be an angle here. Some will have angle here. Aejc. And some will have no angle there. So these are all genetically governed. And the last example I will give you, which will not be that nice to listen to, but still biology is biology, whether you like or not. सब लोगों को कान में earwax होता है ना? Earwax is not a very nice thing to discuss, right? I understand, but biology is biology. So earwax होता है? Now I will ask you, raise your hands. How many have sticky earwax? Raise your hands. Sticky earwax. Sticky means you don't know. With chikku chikku. Sticky earwax means. See? And now put your hands down. Few of them have raised. How many of you have powdery earwax? See? Some. And many of you have not raised your hands, thinking that this is not nice thing to say, right? But I have understood one thing for sure. There is variation. There is a variation. There are some people those who are raising this side. There are some people those who are raising for that side. Means there is variation. Means there are genes which are different. Hello, please. No talking. Now I have successfully made you understand how the things are genetically governed. Many things are genetically governed which you cannot do anything about. Similarly, such a thing is also there in our chromosome number 9. What is? There is one gene here. Its copy is also in the other chromosome, which makes you your type of blood. Means if it is a certain type of gene, you will have A blood. If it is a certain type of gene, you will have B blood group. Am I clear? So it is genetically governed. As one gene is coming from your mother might be having A gene, your father might be having B gene. Since both are of the chromosome number 9, the person will be definitely having A B blood group. Understood? One side A is coming from father, the other one is coming from mother, or vice versa, and you will have AB blood group type, right? If both the genes are AA, then the person will be A. If both the genes are BB, the person will be B. A. What is the word about O?
They can also be overridden because AO, now A and B are dominant over.
Now, come second. Now we are coming to another point called dominant and recessive. Right? What is the meaning of the word dominant and recessive?
There are two alleles, A and B, and A is dominant. Then if a person has both, he will show the characters of A.
Correct. Let us explain to others also.
That's a character.
So what happens is, for A blood group, let the gene be I. Let us give a name to the gene I. Okay? A blood group person, since we are having two chromosomes, and 9th chromosome is also in 2, so we will have to write 2 times I. 2 times I. A blood group possibility is this: in both the chromosome, A gene. So such a person will be definitely A blood group. There is another possibility also. What is that possibility? A and small I. Either you write small I or you may write IO also. Same. Both are same. Am I clear? I null. Yeah, small I means that is recessive. Recessive gene or recessive allele which will not be expressed. So if such a situation is there, A will express, I will not express. So obviously the person will be A type. So either the person will be homozygous A or heterozygous A. This is heterozygous since two different things are there. One is dominant gene, the other one is recessive allele. Recessive, अभी allele का मतलब नहीं समझोगे, so recessive gene से ही चलो, and these are the two dominant genes. ठीक है? Similarly for B, what will be the thing? IB and IB. IB, small I या IO. What about AB? What will be the case in AB? IA. Right. IA. And for O? Small I, or you can write simply IO, whatever you want. That means he does not have the antigen for A or B both.
We can write also as I0 and small I.
That is generally you adopt any one, either this way or that way. Okay? Small I is better to write because it shows that it is recessive. We will come to that. We will come to the crossings also. I guess this slide is clear enough for you now. Read the slide well, and if you have not understood anything, let me know. With the knowledge of chromosome genes, have you understood the slides? Yes. Slide meaning? Autosomes and yes. Okay, fine. Autosome is a nice thing which I missed. Autosomes means 22 chromosomes are called as autosomes. All the 22 chromosomes are autosomes. They are containing all the characters that govern in your body every here. Whereas the last chromosome, 23rd chromosome, is called sex chromosome, which determines the sex of the person. Okay. So 22 pairs of chromosomes are autosome. Last pair of chromosome is called as sex chromosome. If last pair of chromosome is XY, the boy, it will be a boy. If both the chromosomes are XX, then it will be a girl. Got me? So we are not into sex chromosome studies right now. Just it's a 9 chromosome matters, so it is an autosomal condition. So we will consider this as a autosomal character because the blood group is determined by chromosome number 9. Done? Now the very much awaited point. What about that positive negative thing? We have finally come to that rhesus factor. Now this rhesus factor is what you know, Rhd. This is another antigen. Till now we were talking about A antigen and B antigen only. So far we know about these two antigens, right? There is another third antigen also in the same RBC, suppose this is containing A, this contains B, and it may contain another antigen also. So if that antigen B is also there, we call such a person as what? Because he is containing the antigen also, so we will call such a person as Rh positive, rhesus factor positive. Now this rhesus factor was initially discovered in rhesus monkeys. Rhesus monkey is a kind of it's a tribe of monkey, okay, African in origin, and in them it was initially discovered. So we call it as rhesus factor. From the rhesus monkey, we have got this word rhesus factor. So if the D antigen is present, it means the person is positive. If this D antigen is not present in someone, we will call him as AB negative. Here, if this antigen is not there, we will consider that person as AB negative. Done?
And what if we give a person with AB plus
We will come to the RH incompatibility now once we complete this. So RH positive A with RH positive negative, RH positive and RH negative, both the things are compared on the slide. If anybody could not understand anything, let me know. RH antigen is shown in green color. I don't know whether you can understand it or not. A antigen is understandable. With A, there is a green D also. If that green D is there, it is RH positive. If green D is not there, then RH negative. RH antigen नहीं है ये होता भी है. Many people are RH negative, but its percentage is less. In our human beings, especially in this Indian subcontinent part, we have around 80 to 85% people are RH positive. Around 15% people are RH negative. They don't have RH antigen. That is not a problem. Whether antigen is present or not present does not make anybody good or bad. It is a kind of type. It is a type. Okay? Yeah.
Someone who is RH negative
We are coming to the RH compatibility factors. We are coming. Now here.
I want to say that in every RH positive blood types, there is always an RH antigen.
Yes. RHB antigen has to be present, then only we will call him RH positive.
And where it is not present,
Then RH negative. Right? Now coming to the point of compatibility based transfusion. And next slide. Okay. As I said, 85% of the population is RH positive and the 15% of it is RH negative. We have discussed it already. Next slide. Now, as I was telling you a few moments back about agglutination reaction, do you remember antigen-antibody reaction to happens and the blood curdles? This is the curdled blood. If a wrong blood comes, suppose if A blood group person gives blood to B blood group person, this is the type of blood situation will happen in the body. This is called agglutination reaction. We have already discussed it. Why it happens? Because A antigen will be attacked by the A antibody. And since A antigen antibody recognition will happen and attacking will happen, what will happen? The blood will curdle up and the blood cells will be destroyed. This is the destroyed blood cell. If you do you all want to see it yourself by your own blood? Let's do it tomorrow. What do you say? But I might not be able to give everybody a chance. We will try to give 4 or 5 people a chance to see for their own on their own whether it really happens or not. And if you don't know your blood group, I would like you to have that child first. If you know your blood group, there is no maza. So सबको पता है? कोई है जिसको नहीं पता है? और कंफ्यूजन है? तो हम कल इसके ब्लड से करेंगे ना? ठीक है? So we will see 4 or 5 volunteers we will take and we will give you the chance to see for yourself how agglutination reaction happens. You will see it yourself. Yes, fine. Settle down. Next. You can see now agglutination in anti-A blood group. You can see anti-A and anti-B. Anti-A and anti-B, both the bloods are given here to show you how the blood group A will react. Anti-A, anti-B refers to the antibodies. Say for example, a person is having A blood group. The first line is of A blood group. Okay? That person is having A. How do we know? Because when we gave that person A antibody, the blood has coagulated. The blood showed agglutination. So we have understood that person is having A blood group. Okay. Similarly, for the person who is having B blood group, how do we know that he is B blood group? When we give him B antibody, his blood group has coagulated. His blood has coagulated. Understood? Similarly, when a person is O blood group, his blood has not coagulated at all. Because why? We have given both the antibodies, A antibody भी दिया, B antibody भी दिया. लेकिन उसका blood coagulate हुआ ही नहीं क्योंकि उसके पास कोई एंटीजन ही नहीं है. That is why he is blood group O. And AB blood group वाला इंसान के दोनों ही AB पे coagulation होगा. जब antibody A दिया, antibody B दिया, दोनों में ही what happened? The blood coagulated. Understood? Anybody with this picture you have clearly understood. If you want to do it tomorrow, we will make you do also. ठीक है? 5 people will be getting chance to do. Chalo, every batch we will give 1 or 2% chance. Next, this is the same thing which we have discussed so far. A blood group with A antigen will have B antibody and hence it is called as उसका donor group क्या है वो भी लिख दिया कि वो कहाँ से blood ले सकता है या दे सकता है. So we have given this chart. I think you can understand the meaning of this chart. If you fail to understand the meaning of this chart, please let me know. Next one. This is a chart, overall chart, which shows who can give blood to whom. Have a look at it. With your knowledge now, understanding now, you can understand the meaning of this. Sit down. B antigen के लिए एक और D antibody रहता है. Antibody for D is also available. So we put that D antibody into the blood. If the blood coagulates, at least he is Rh positive. And the other one is for D. We will get it done tomorrow for D also. You will come to know whether you are positive or negative also.
There is a incompatibility. What will happen to the person when he dies?
Yes. What happens now is Rh incompatibility reaction. AB minus means AB negative. You want to say yes. AB negative can take blood from O negative. O negative being the universal donor. मैंने वहाँ पर blank रखा है क्योंकि it was written yes, which was wrong. So it is basically we cannot take blood from AB positive. Any AB negative, any negative Rh negative person cannot take from Rh positive. I'm not understanding. AB negative can take from A negative. AB negative can take from A negative because AB negative से ले सकता है. Yes, that yes should be over there, right? Correct. That means you have all understood. I'm happy that you have understood. That is why you must make that connection. A minus can give blood to A plus. Yes, minus can give, but minus cannot take. Right?
Some people are having blood groups like A1 positive, A1 B, then what?
A1.
A1 positive.
A1 positive. I have never heard of it. Sorry, can you give me some document related to that? Anything else? Yes, please. Fast, please. Excuse me, can you all please stop talking? Yes. O negative. See here. You can verify from the chart.
So they are universal donor.
O negative can take only from O negative. O being negative cannot take from positive.
So negative.
I will see this document and all that.
So O negative is the universal donor.
Huh?
O negative is the universal donor.
O positive is a universal donor.
But here,
O negative can take from O negative only.
O negative can also give to any group. But some O positive cannot give to rhesus factor negative.
See, being O negative now, he is not having the antigen. Okay, he cannot take from O any positive person because then he will receive the antigens. Any negative person cannot take from positive. That I am repeating again and again. Please remember also, if you want to write it down in your notebook. Any negative person, Rh negative person cannot take from even the same blood group also. He cannot take from positive person.
Take what has happened.
Then I same thing, agglutination. Yes. Yes, the same. Finally the thing is time. Yes, he will.
Now, how does this depend on genetics? Because my mother and father are both positive, but my elder brother is O negative.
No, it is not possible. See, I'll tell you why. Wait. There might be somewhere some error. Because this is genetically covered, I would like to see the medical documents related to it. Otherwise, just saying the documents. Because, hello, please. No talking, please. This kind of, it's not first time that I'm getting this. And when I checked for myself, a child who came and said, yeah, I'm so and so, even his identity card was saying that, but when we tested it, did not comply with it.
There might be errors.
So it is very common. Such errors are common. Hello, please, no talking. Just like this. Could you please be quiet? Otherwise, I will not be able to move on it in my session. Please. Now, Rh incompatibility has more, of things to be learned than simply blood transfusion. See, Rh incompatibility is beyond what you are thinking right now. About being Rh positive can give blood to negative or negative can give to positive. ये छोटी सी चीज है. इससे भी बहुत बड़ा चीज आपको जानना चाहिए. So please be patient. Don't be impatient and don't talk amongst yourselves. That is not a good thing. So, Rh incompatibility for a case where mother is bearing her child with mismatched Rh.
Secondary.
Then what will be the situation? That is more important topic than simple blood transfusion. So let us begin with that. Next slide. A person with Rh negative blood can develop Rh antibodies in the blood plasma if he or she receives blood from a person with Rh positive blood. अभी मैंने वही बार-बार आप लोगों को बोला था, a person with Rh negative cannot take blood from Rh positive, right? So a person with Rh positive blood can receive blood from Rh negative, however. So next slide. This is a very important slide which I was about to tell you. This Rh incompatibility goes beyond the simple blood transfusion related issue. What is it? If a mother is Rh positive, if a mother is Rh negative and the child she bears is Rh positive because the father is Rh positive, then the child might be Rh positive. In that case, what will be the possible condition? As you know, the Rh factor which is present in again the blood cells, the blood cells are generally not mixing in the placenta. For your information, the mothers and the child placenta only interdigitate. What is the meaning of the word interdigitation? Means they will come closer to each other but they will not mix. Okay? So that is why a mother and the child can have separate blood groups, separate Rh levels also, Rh groups also. But in case if the mother is Rh negative and she gets a child who is Rh positive, for the first child there might not be any bad situation, unpleasant situation, perhaps. But for the second child, there will be certainly many issues, okay, unless medically intervened, unless medically seen. So what we have seen in this slide, let us understand. Rh positive father, Rh negative mother, and mother being Rh negative, now she gets a child who is Rh negative, Rh positive. Okay? Rh negative mother with Rh positive baby. This is the situation. Now what will happen? Rh positive baby's blood cells enter mother's bloodstream. When this can happen? Not right in the first, second, and third trimester. No. Only during the parturition, that is the process of delivery, there might be placental breakage and some chances of mixing of child's blood with the mother's blood. Right? At that time, because the child is a small one, if you compare like some hours, some few hours, so what will happen? The child's blood will be not destroyed, but since child is anyhow Rh positive, no issues. But mother, what could be her case? Mother is lacking in Rh antigen. So what will happen? She will develop, if she gets some fetal Rh positive cells in her blood, so she will develop antibodies against those Rh positive blood cells. So mother will have Rh antibodies against fetal Rh antigen. Okay? So what will happen? Mother will keep the antibodies in her memory cells. Your immune system has got memory cells. This interaction will be recorded and will be saved in mother's memory cells. In her immune system, in the mother's immune system, there are some memory cells which will keep this interaction of the child's fetal Rhb antigen and mother's Rh antibody interaction. Some cells will be interacting, agglutination hoga. Those will be recorded and saved in the memory cells of mother. First child will be born safe, क्योंकि mother का जो ये agglutination reaction है, it is not going to be havoc on the first child. It will not be killing the first child. First child will go unhindered, undamaged. But what about the second child? Second child, when the second child is also Rh positive with the same Rh negative mother, now what will happen? Her pre-recorded and pre-saved memory cells will trigger in the first trimester only. First trimester means first, second, third month only. She will start giving her this pre-saved agglutination reaction. Uske body mein already saved है. Now she will do what? She will produce बहुत ज़्यादा amount of Rh antibodies. Many Rh antibodies she will produce within first, second, third month only. Because she has her memory cells of the previous interaction. Now those antibodies will go and start attacking the Rh antigen of the baby. Because baby is Rh positive. The baby is having Rh antigen. Those mother's antibodies will now go start attacking the baby's antigen. And the baby will have serious repercussion for this. And as a result, we call such a situation as next slide. Yes. Next slide.
But you told that they will not be connected to
Only I said in the first delivery process. During the delivery, placenta will break. Some fetal cell will go into the mother's body because of the breakage of the placenta. Second pregnant mother is having the pre-recorded antibodies because her body in the first delivery process has recorded the activated antibody reaction in the memory cell. But the first child is out, so she could not destroy the first child. First child is already given birth during that delivery process. First child went unhindered. But second child when comes, her pre-recorded saved file है ना, अभी उसके पास saved file है. heart is built, immune system built. So that same file will start triggering, making many antibodies. Understood now? So this is the whole thing which I have told in a flow chart. Have a look, please. Have a look at the entire flow chart. You will understand each and every point. Whatever I said, match it. First pregnancy, mother Rh negative, fetus is Rh positive, and there is no problem in the first one. Next pregnancy, the mother is Rh negative, the child is Rh positive. Then what will happen? Rapid production of immunoglobulin G. Immunoglobulin G is an antibody against antigen, Rh antigen. Okay, so that will cross the placenta in the first trimester itself and will start killing the fetal cells. It will be either, unless medically intervened. Doctor, if they come in between and they give some, different kind of, medication and all, that is a different case. But in many times when we see that the mother is not even aware that she is negative, Rh negative, and she is going for the first child the janam le liya अच्छे से, she is okay, she will not even know the second child will be Rh positive and she will be unknowingly killing the fetus in the first trimester only. This happens in rural India where people are not much aware about their blood and all that. So this case is called as, next slide. This condition is called as hydrox fetalis or erythroblastosis fetalis. This condition is called as hydroxis fetalis or erythroblastosis fetalis. Next one. This is a case where I don't want to put it for a long time, not very pleasant to see. So this is again not much different. Sorry.
What is the disease?
Huh?
The
The disease name I'm writing. Like this name. Erythroblastosis fetalis. This is also in short form called as HDM. Hemodialytic disease for newborn. So it is erythroblastosis fetalis. You just write down erythroblastosis fetalis. That is more than enough. Otherwise, hydroxis fetalis. Write down this one. Erythroblastosis fetalis. Erythrocytes will be breaking down. The child will be having swollen stomach with full of liver condition will be bad. This kind of things, which I don't want to explain much. And what is your point? Could not hear you.
So now for EDF,
What? HDN का full form बोल रहे हो? Hemodialytic disease of newborn. Forget. Write the one which I have written. I'm loving it. बहुत ज़्यादा नाम होगा तो confusion करना. Then there is no problem. No problem. Fetus negative होने से कोई problem नहीं है. Rh positive mother के साथ negative fetus का कोई problem. Only this problem is with that. We have discussed this next week. Now we will be discussing some genetic process and we will complete our lecture today. After, say for example, we will do some process. Okay, I'll give you some process to do. Say for example, the father this is the sign of a male. In biology, we give the male as this sign, and the mother is this sign. Okay? You will remember? Okay. If, say for example, mother is A blood group and father is B, say for example. Okay? Now, we will have for A blood group, what all are the possibilities? Right? Right? Yes or no? And for B, what are the possibilities? Right? Let us consider a cross. You might be having many people will be knowing their parents' blood group also, right or not? Yes. Let us understand. Consider a father is having A blood group, and say for example, we know that it is this, and a mother is having this group, we know that it is this. How to cross it? You will have to cross it just simply like this. IA, IB, IA, IB. All the children will be having what blood group? All the children will be having what blood group? AB. Every child will be AB. If that is the case, then we will understand the mother and the father were having this particular blood type. Okay? But consider this case. If we are not doing it like this, for example, if we are thinking, let us consider this. If mother is IA and father is that combination, what will be the case? IA, IB, IA, small I. Similarly, IA, IB, IA, small I. Am I right? So in such a situation, 50% children will be having AB blood group and 50% will be having A blood group. If that is the condition, you see that you can make out that parents were not IA, IB. Then the parent is IA, and IB. Am I clear? Now, let us just randomly say, make a genetic cross from you people, those who know about your parents' blood type, and yourself also. Anybody who knows parents' blood type? Both the parents, okay? Fine. I mean, I will give him the chance because he is near to me. I can hear him well. So, louder, please. Your father is O. So, what should be the case? This. Only possibility. And mother is A. So mother is A. Right? So if this is A, then we have both the possibilities of this. Either your mother is this or your mother is this. Right? Let us now understand where you fall. Okay? IA, small I. Correct? IA, small I. IA, small I. IA, small I. If this is the case, then you will be, all the children will be, every time the child is born, the child will be A blood group, right? And if your mother is into this category, then what? The percentage, your 100% children will be A, right? Always it is A only. But if this is the case,
70%.
please don't talk because next time you will be asked to come here and do the class.
75% will be A.
Now let's see. We are doing this with this now. IA, small I. Yes. Small I. This is IA, small I, and this one will be small I. So we see 50% children will be A blood group and 50% will be O blood group. So where do you fall? You are A. You could be of this or you could be this. Your brother is O, then your mother is definitely this. There is no other option. Your mother can never be IA. Your mother has to be IA, small I. Clear? You can go and tell your mother. Understood? Because it's good that his brother is O, so we can now know that he is, your see if we can understand this cross now. If you don't understand, let me know. That group B, both are IB small I. And this is another way to do the same thing, but we are not going by correct square, we are going by the upper one. It is easy to make out. So please note up, if parents are B, both are B, and both are heterozygous B, means IB small I, then the possibility of the children will be 25% children will be homozygous B, 50% children will be heterozygous B, and rest 25% children will be O. Is there anybody who could not understand? Now, if I give you some process to do, please do it in your notebook and let me know the result. Take a cross where mother is. I'll write it on the board and do it yourself. Father is O, mother is AB. Written. Do the cross. Now don't see each other's notebook, do it yourself. Give me the results. Yeah. Why so much of time? It's very easy. Do it fast. Yes. Huh? How much? 50%? A. And 50% B. See the beauty of the science here. None of the parents were A or B. None of the parents were A or B. But all the children are A or B. Parents were either O or AB. But children are either A or B. None of the children are having the blood group same as parents. Right or not? And now you are expert in making the process. Now you cannot say and tell me कि ऐसा ऐसा है, मम्मी का ऐसा है, डैडी का ऐसा है, मेरा ऐसा है. So I will not agree to it. It's all genetics, it's all science. You cannot come up logically against science. There might be some error somewhere, we can sort it out, but science is science, we have to accept it. Anybody who has not understood? अच्छा, मेरे को पहले बताओ कि किसी ने ये कर नहीं पाया ऐसा है कोई? Is there anybody who could not make that cross? Raise your hand, I will help you. Because see, tomorrow there will be an exam, a test. So if you have not understood, it is better we sort out now. Tell me, you could not do? Fine, let me see. Why you could not do? So one child has told that he could not do. I guess there will be a few more for them. For that, first I want all of you to be quiet. O means only possibility, right? AB means only possibility, right? Yes or no? Yes. So let us do it. Right? So what is this? A, B. Simple, 50% A, 50% B. No O, no AB. As parents. So ये हो सकता है कि पेरेंट का A B है, पेरेंट का O है, बट बच्चे लोगों का उनके साथ मैच नहीं है. इसका मतलब ये नहीं कि बच्चे उनके नहीं हैं. So there lies the beauty of genetics. Yes. Please keep quiet. Yes.
Every 7 years, a person's allergies have a chance of disappearing.
Allergies. Okay. Allergies develop with age. Allergies go away with age. These are all basically, allergy is basically an immunological reaction which is because of one antibody called IgE. Immunoglobulin. अभी मैंने तुमको थोड़ी देर पहले देखा था ना IgG that can cross the placenta, that Rh antibody, right? Similarly, this is another antibody IgE. There are 5 types of antibodies: IgD, E, M, A, G. Or you can just simply remember DMAG. DEMAG, DMAG. There are 5 antibodies in the body of any animal, any mammal. Okay. So IgE is the allergy-causing antibody. Allergy is also a kind of an immunological reaction. Allergy is not bad. Allergy means like fever. Fever is also an immunological reaction. That something has gone inside you. So you are reacting to it, immunologically reacting to it. Similarly, IgE is also something that has gone inside you for which you are reacting. Some foreign element has come to your body, you are reacting to it. Your immunology is reacting to it. So it is producing IgE. So that your mast cells come to that place and start doing some work. You are calling your mast cells. Yes, there are some specific things which we are allergic to, which we know our body doesn't have, way to react with, way to combat, way to nullify. So we react with it.
Where is IgD found?
IgD, all the immunoglobulins are found in the blood. All the immunoglobulins, all the antibodies are found in blood.
IgD is used if for placenta.
Ish.
IgD.
I'm sorry, I'm not able to hear you well. We are running short of time. Just wrap up.
IgD is for like allergy.
Allergy.
So what is the use of IgD?
IgA. E.
D.
D is IgD. E has got some other stuff. I cannot really recollect right now. Every antibody has got separate function. Anything else? Slide next one. We can check. I think this is the last slide, or maybe this was our last topic. How to do the genetic process. We are done with our topic today. I am happy that you have heard me well. Patiently, you have tried to learn from me. I'm really happy for that. If I could not answer your queries fruitfully or successfully, let me know. I'll try my level best next time. Again, we will be meeting tomorrow. Come up with more questions. I will be happy to answer them. With that, we can come to the end of the session. Thank you.
I deem it only fitting that we give her an exit, a worthy exit for the class that she took. My name is Suri Chaudhary and I am from Aejc Skyline. Now, biology is often confused as a subject that's not clearly understood. People think it's boring. Half of the children sleep in the middle of the class. I was really happy and really surprised to see how engaging the biology class was and how quickly and swiftly she completed the class and how clear she was with her answers. Even if it was off topic, she was ready. She was okay. You want to know, I will tell you. I want to thank you very sincerely, ma'am.
You made my day. Thank you so much.
Children, we can have 15 minutes break. Please go out of the hall and bring some small snacks. 15 minutes, within 15 minutes, you have to come back and take your seat, okay? Don't delay on this.
मला वाटलं आता 15 मिनिटच ब्रेक आहे. आपण बोलतो ते सगळं ऐकायला नाही. आता ऑफ मोड मध्ये गेले. ऑन झालं नाही वाटत. ते थोडं आता 1 मिनिट घेऊ. मी जाऊ वॉशरूमला. हो. ये ऑफ हो गया है, ये भी ऑन नहीं होगा. अच्छा ऑन करो. अच्छा देखना है ना. एक्चुअली इसका सिस्टम भी रखना चाहिए था. खाली कीबोर्ड रख दिया है. वही करना है. मैंने डायरेक्ट प्रोजेक्टर ऑफ
ये वाली कोड नहीं चाहिए, सिर्फ ये वाली कोड